All right, so this last video is just going to be talking about the last macromolecule, which is on nucleic acids. And the big thing that you want to remember about nucleic acids is their function. And then there's really three main nucleic acids you should be aware of. First one being DNA, but there's also RNA, and then don't forget ATP is also made up of nucleic acids as well. So um, in terms of that, you also want to kind of understand that, like, if you look at a human karyotype, each one of those lines represents a gene, and each one of these realistically is made up of individual nucleic acids. So, kind of the roles of what they are, like I mentioned earlier, the most common thing that you guys should be familiarized with is these two. Your nucleic acids are made up of, it makes up DNA and RNA, depending on the sugar that is there. So, um, as we kind of have already covered now, DNA replication, you want to understand that it just shows that you have that replication fork. It goes in both directions. The DNA, the whole point of this is DNA is read to make mRNA. That mRNA is then read to make proteins. So just to remind you that protein synthesis occurs on the ribosomes. So nice little simple overview picture here from your text is that here's DNA. DNA is opened up and you read a section of it called a gene, and that makes your messenger RNA. If it's eukaryotic, that mRNA leaves, goes out to the ribosome, and then this is the building of your polypeptide. So the last video talked about how this polypeptide is going to go on to fold and do its function. So those are the amino acids they build to make that polypeptide. These ribbon structures, those are your nucleic acids. So if you look at the actual components of a nucleic acid, there's three main parts you want to know. This is going to be your phosphate group right here. This is your sugar. Each one of those kinks represents a carbon. So I have carbon 1, carbon 2, carbon 3, 4, and then there's your fifth one held at an oxygen right there. And then this is your base. So the difference between these nucleic acids, like adenine, thymine, cytosine, and guanine, that's what changes. So in terms of the big components of this, you just want to know the basic three components. You get that phosphate, the sugar, and then you have the base. Now, in terms of this, you don't have to know the drawing rule by any means of how to draw each one of these, but you do want to know the difference between a purine and a pyrimidine. So purines are adenine and guanine. Remember that pure as gold, A for adenine and G for guanine. And then the pyrimidines are going to be cytosine, thymine, and uracil. So those are going to be the single ring structure. The purines, adenine, and guanine have two ring structures. So um, this shows you the difference between the sugar. This is showing you that if it's deoxyribose, you're short one hydroxide here, whereas if it is that ribose sugar, it has that OH here. So thinking back here to this picture, just remind you that if there is an OH here, we're going to call that ribonucleic acid, but if it's missing, that's what we call it deoxyribose. So that's the sugar for deoxyribose. And like I mentioned previously, this, when we say nitrogenous base, this is any one of these five bases here. And so this kind of is a revisitation of how these things get structured. They're held together by these covalent bonds. Always remember that if there's the free hydroxide, that's typically your 3' end. And if there's a phosphate with nothing attached to it, that's your 5' end or your 5' carbon. So just kind of a nice little overview of DNA versus RNA, the two main categories of these nucleic acids you should know. The DNA is found in the nucleus. If it is eukaryotic, remember prokaryotes, they still have DNA, but there's no nucleus, so they're just in the cytosol. Double helical structure, deoxyribose is the sugar involved. The bases are adenine, guanine, cytosine, and thymine. RNA has uracil instead. It has the ribose sugar, so it's short that one hydroxide. It's always going to be single-stranded. And DNA runs anti-parallel, so remember it goes in reverse directions because it is that double helical structure. So nice little drawing here showing you the double helix. C pairs with G, A pairs with T. We get into this in chapter 16, but remember these are held together by hydrogen bonds. So this would be called the backbone. So just remember that, and then here's your bases. So in terms of that, that is kind of a nice little overview of what these nucleic acids are and their main function.